Welcome back to the deep dive. Today we are wading into what I can really only describe as a biological war zone.
Yeah, waiting is definitely the right word for it.
Right, because what we're looking at today, it lives in the water in the soil, and disturbingly often inside of us. We are tackling parasitology.
And it's a topic that, well, it triggers a very primal reaction, I think.
Oh, absolutely. The ick factor is real.
It is. You talk to people about bacteria and they nod. You mention viruses and they get a bit anxious about contagions. But you talk about worms living in the intestine or amoebas eating brain tissue and people physically shudder.
Yeah, it feels invasive in a way that a virus just doesn't.
Exactly.
But we do need to push past that today because we have a massive stack of research to get through. We are anchoring this deep dive primarily on Murray's Medical Microbiology, Chapter 67.
Right, and we'll be pulling in some crucial clinical color from Japin's Chapter 46 as well.
And the mission here for you listening, it isn't just to list a bunch of creepy crawlies.
No, listing them without a system is entirely useless. If you just try to memorize 100 different bugs, you will fail the exam. And more importantly, you're going to miss the diagnosis in the clinic.
So our goal is to build the taxonomic scaffolding.
The mental shelves.
Exactly, the shelves. So that when you encounter a specific parasite in your practice or your studies later, you know exactly where it fits in the grand scheme of life. We are organizing the library before we start reading the books.
And we really need to establish the stakes immediately here. Because there's this tendency, especially in modern Western medicine, to view parasitology as a quote-unquote tropical issue.
Something that happens over there.
Right, which is incredibly dangerous.
I was actually reading the intro to Murray, Chapter 67, and it hit me with this stat right out of the gate. Malaria alone, just malaria, it causes over 500 million infections and millions of deaths every single year.
It's staggering. But that tropical label is obsolete. We live in a world of hypertravel, of refugee migration, and crucially, immunosuppression.
Because of HIV transplants, chemotherapy?
Yes, exactly. For these patients, an organism that might just be a nuisance to a healthy person, it becomes a lethal threat. So parasitology is global medicine now. It's happening in London, New York, Tokyo, not just the rainforest.
Okay, so let's start at the very top. The 30,000-foot view. We are officially leaving the world of bacteria.
We are in the eukaryotic domain now.
Right, and this is the single most important distinction when it comes to treatment.
It really is. Bacteria are prokaryotes, so they're structurally very different from us. They have cell walls we don't. They have completely different ribosomes.
Making it relatively easy to find a drug that kills them but ignores human cells.
Selective toxicity. But parasites, they are eukaryotes. They have a nucleus. They have mitochondria organelles. Biologically speaking, a parasite is our cousin.
It's way more similar to a human cell than a bacterium is.
Which means a drug that poisons the parasite has a much higher chance of poisoning the patient.
So the margin for error is razor thin?
Exactly. That is the central challenge of anti-parasitic pharmacology.
Yeah.
You're trying to kill a eukaryotic cell inside another eukaryotic organism. So as we go through this, you have to keep in mind that the biology of the parasite dictates the toxicity of the treatment.
Now, within this whole world of eukaryotic parasites, we have two massive kingdoms to contend with, the Protozoa and the metazoa.
The single-celled versus the multicellular.
I want to give you a visual anchor here, a mental image to come back to. When we talk about Protozoa, what should they be picturing?
Picture a drop of pond water.
A drop of water, okay.
Yeah, in that drop, you have these tiny, microscopic, single-celled organisms darting around. They're complex, right? They do everything an animal does, eat, move, or reproduce.
Yeah.
But they do it all within the confines of a single cell. That is the protozoa.
And the metazoa.
Picture a plate of spaghetti.
I love that.
Or a snake, if you prefer. These are animals, multicellular. They have tissues, organs, nervous systems. These are the Helminths, which is just the scientific term for worms, and the Arthropods, the bugs that carry them.
So microscopic. You can often see them with the naked eye.
Often, yes.
Okay, so the drop of water and the plate of spaghetti. Let's dive into the drop of water first. The protozoa.
The unicellular operators.
Now, I remember my old textbooks classifying these things very functionally. It was like, this one has a tail, this one has a false foot, this one doesn't move.
Yeah, the morphological classification.
But Murray throws a bit of a curveball. He talks about supergroups.
Science moves on. That old classification, it was intuitive, but it was evolutionarily wrong. The new taxonomy that Murray emphasizes is based on genetic lineages, specifically ribosomal RNA sequences.
This feels a bit like when Pluto got demoted.
It does a bit. It's necessary, but it can be confusing. But it's high yield for modern exams. Understanding the genetic lineage tells us about their biochemistry, which tells us about drug targets.
So we have to grapple with the supergroups. Let's unpack the first one. Supergroup number 1, excavata.
The excavators.
Sounds like heavy machinery.
It refers to a groove on the surface of the cell, a feeding groove that looks like it was excavated right out of the organism. But the real headline here is that this group contains some of the most ancient eukaryotes on the planet.
And for medical purposes, we break this down into 2 main clades or branches. The first is Metamonada.
This is where the old-school flagellates live. The big stars here are Giardia and Trichomonas.
Let's start with Giardia. I think anyone who hikes knows about Giardia.
They definitely should. Giardia lambelia or duodenalis, depending on the nomenclature. It's the classic camping nightmare, the cause of beaver fever.
Structurally, what makes Giardia unique in this group?
Well, it's a biological oddity. Under a microscope, the Giardia trophozoite literally looks like a little face staring back at you. It has 2 nuclei that look like eyes and flagella that look like hair.
A little ghostly face.
But the most critical structure that Murray highlights is the suction disc.
A suction disc, like a plunger.
Essentially, yes, it's on the ventral surface, the belly. It uses this massive suction cup to clamp onto the villi of your small intestine.
So it's holding on against the flow of the gut.
Exactly. And here is the key pathology. It doesn't invade the tissue. It doesn't eat your blood. It just carpets the intestine. It covers the lining like a tarp.
And if you put a tarp down on the gut lining, you can't absorb food.
Precisely. You get malabsorption. That's why the clinical presentation is so specific. Foul-smelling, greasy floating stools. Steatorrhea. Because the fat isn't being absorbed, it's just passing right through.
Greasy stool equals Giardia. Got it. Let's compare that to its cousin in this group, Trichomonas.
Trichomonas vaginalis. As the name implies, this is a pathogen of the genitourinary tract. Structurally, it has flagella, but it also has this weird undulating membrane, like a fin running down its side.
Now, there is a crucial difference between these two regarding how they survive outside the body, and this feels like a major exam point.
It is absolute gold for exams. It comes down to the cyst. This is the key insight for this whole section, really. Most intestinal protozoa alternate between 2 forms, the trophozoite and the cyst.
Let's define those for the listeners. I like to think of the trophozoite as the soldier.
That works perfectly. The trophozoite is the active form. It's feeding, dividing, causing the symptoms. But like a soldier on the battlefield without armor, it's fragile. Stomach acid kills it. Drying out kills it.
So the cyst is the bunker, the organism wrapped in a tank.
Exactly. When the trophozoite moves down the bowel and starts to dehydrate, it balls up, secretes a thick wall, and completely shuts down its metabolism, becomes a cyst.
And that's the infectious form.
Right. It can survive in cold water and soil, even in chlorinated pools.
So Giardia forms cysts.
Yes. That is how you catch it. You drink the cyst from a stream. It survives, your stomach acid gets to the small intestine, and the bile salts trigger excistation.
The bunker opens and the soldiers come out.
Exactly. But Trichomonas.
Trichomonas does not form cysts, right?
Correct. It only exists as the fragile feeding trophozoite.
So if it's that fragile, how does it get from person to person?
Direct contact only. It cannot survive the environment. That's why it is strictly a sexually transmitted infection. If Trichomonas fell on a toilet seat, it would die very quickly. Giardia could wait there for weeks.
That is a perfect example of how structure dictates epidemiology. No cyst means direct contact only.
Precisely.
Okay, moving to the second clade in the excavator group, the Disecristata.
A mouthful of a name. It refers to the shape of their mitochondria. They have disc-shaped cristae. In this group, we have some heavy hitters. Naegleria leishmania and Trypanosoma.
Naegleria, that's the brain-eating amoeba, right?
That is the one, Naegleria fowleri. Interestingly, it is actually a free-living amoeba. It's not actively looking for humans. It just hangs out in warm, freshwater eating bacteria.
But if a swimmer gets water forced up their nose...
It crosses the cribriform plate, it follows the olfactory nerve straight into the brain, and it releases enzymes that literally liquefy brain tissue.
Primary amoebic meningoencephalitis.
Yes, it is nearly 100% fatal.
Terrifying. But let's look at the others. Leishmania and Trypanosoma. These are grouped together as hemoflagellates.
Meaning they are flagellates that live in the blood and tissue. And unlike Giardia, they don't rely on you drinking dirty water. They rely on vectors.
The insect taxi service.
Exactly. And matching the bug to the insect is a classic board question game. So let's run through the matchup from Javits.
Let's do it. Trypanosoma brucei. This causes African sleeping sickness. The vector is...
The tsetse fly. And remember, the tsetse fly bite is painful. It's not subtle at all. It's like getting stabbed with a hot needle.
Next up, Trypanosoma cruzi causes Chagas disease in the Americas.
The vector here is the reduvid bug, also known as the kissing bug.
Sounds romantic, but I'm guessing it's not.
Not even slightly. It bites you on the face, usually around the lips or eyes while you sleep. But here's the gross detail that matters from the text. The transmission isn't actually in the bite.
Wait,
The bug defecates while it eats. The parasite is in the feces. You wake up, you scratch the itchy bite, and you manually rub the infected feces into the wound or into your eye.
oh man, that is horrifyingly specific. Scratching poop into your own wound.
It's effective, though.
Clearly. And finally, leishmania.
Transmitted by the sandfly, a tiny silent flyer.
Okay, so that covers the excavators. Let's shift gears to supergroup number 2, the amoebozoa, the shapeshifters.
This is what most people picture when they think of a germ under a microscope, a blob that moves by streaming its own body forward. Those extensions are called pseudopodia, false feet.
And their feeding mechanism is phagocytosis.
Right. They literally hug their food, whether that's bacteria or your cells, and they just engulf them.
The big bad wolf in this group has to be Entamoeba.
Specifically, Entamoeba histolytica. The name tells you exactly what it does. Histo means tissue, lytica means breaking. It's a tissue breaker.
Javets gives us some very specific clinical images for this one.
It does. In the colon, Entamoeba histolytica creates these classic flask-shaped ulcers.
Flask-shaped, so like a narrow neck at the top.
Exactly. A narrow neck opening into a wide, eroded cavern underneath the mucosal surface.
And if it gets out of the gut?
It loves the liver.
Oh.
It travels via the portal vein and creates an abscess filled with necrotic liquefied tissue that is famously described as looking like anchovy paste.
Flask-shaped ulcers and anchovy paste. Why does pathology always have to ruin food for us?
It's a gift, truly. But there is a trap here for the unwary student that Murray points out, something called Entamoeba dispar.
The decoy.
Right. Under a microscope, Entamoeba dispar looks identical to the killer Entamoeba histolytica. Same size, same shape, same nucleus. But dispar is entirely non-pathogenic. It doesn't hurt you at all.
So if you're looking at a stool sample and you see an amoeba, how do you know if you need to panic?
Well, modern labs use specific surface antigens, or PCR, for DNA. But historically, if you're looking at a slide, there is one visual clue. If you see the amoeba literally eating red blood cells inside itself.
Erythrophagocytosis.
Yes, that is the smoking gun for histolytica.
Yeah.
Dispar doesn't eat blood.
Okay, really good differentiator to remember. Now let's move to the third supergroup, SAR.
Right, SAR. It stands for Streptopiles, alveolates, and Rosaria. It's this huge complex grouping based on very deep genetic links.
Let's break down the clades within SAR. The massive one for us is Apicomplexa.
Formerly known as Sporozoa. This includes the absolute heavyweights, Plasmodium, which causes malaria, Toxoplasma cryptosporidium, and Babesia.
Why the name Apicomplexa?
It refers to the apical complex. Picture the parasite as a cell shaped somewhat like a bullet. At one tip, the apex, it has this specialized structure, a collection of organelles that acts almost like a drill bit or a breaching charge.
A breaching charge to get into human cells.
Yes, they use this complex to chemically and mechanically drill their way into a host cell. These are obligate intracellular parasites. They don't just hang out in the blood, they break into your cells to hide and replicate.
And their life cycles are famous for being incredibly complicated. We always see those textbook charts with arrows going absolutely everywhere.
We can simplify it, though. They usually alternate between two distinct types of reproduction, schizogony and sporogony.
Let's unpack those terms because they show up constantly.
Schizogony is asexual reproduction. It's making a clone army. The nucleus divides multiple times inside the host cell, and then boom, the cell bursts, releasing a swarm of clones.
And this is what's happening in your blood during a malaria fever spike, right?
Exactly. The synchronized bursting of red blood cells is what causes the fever.
And the other one? Sporogony.
Sporogony is sexual reproduction. It involves the fusion of gametes. This usually happens in the vector, so like the mosquito in the case of malaria, it's genetic shuffling.
So clone army in the human for rapid expansion, genetic shuffling in the mosquito for diversity.
Exactly.
Also in the SAR supergroup, we have the Ciliophora, the ciliates.
This one is easy because there's only one human parasite in this entire group. Neobellentidium coli, formerly just Bellentidium.
What's its gimmick? It's covered in cilia. Tiny oars that let it swim rapidly. And it's huge. It has this massive kidney bean-shaped macronucleus that you can spot from across the room, microscopically speaking. It's mostly a pig parasite, but it can definitely hit humans.
And finally, in SAR, the Streptopiles.
Which brings us to Blastocystis. This organism has had an identity crisis for decades. We used to think it was a yeast, then a protozoan. Now genetically we know it's a stranded pile related to algae, oddly enough.
Is that actually dangerous?
That is highly controversial. Many people carry it with zero symptoms. But in large numbers, it can cause diarrhea. It's often considered an opportunist or a commensal with an attitude.
All right, that wraps up our tour of the protozoa, the drop of water. But before we cross over to the worms, let's talk about protozoan physiology a bit more. We mentioned the trophozoite versus the cyst with Giardia, but that applies broadly, right?
Yes, the two-state solution for survival. But there's another physiological concept Murray emphasizes that is absolutely crucial: immune evasion.
Specifically with the trypanosomes. Yes. Trypanosoma brucei, the sleeping sickness parasite, is an absolute master of disguise. Think about it, it floats freely in the blood. The immune system should see it easily.
And it does see it right.
It does. The body makes massive amounts of antibodies against the parasite's surface coat. The antibodies lock on trigger complement and kill 99% of the parasites.
So why doesn't the patient just get cured?
Because the remaining 1% have already switched their coat. They have a library of over 1,000 genes for their surface proteins, variable surface glycoproteins, or VSGs.
So just as the immune system is about to win, the parasite swaps the protein.
Exactly. It essentially goes into the closet and puts on a different jacket. And the immune system says, wait, I don't know who this guy is, and has to start the whole process all over again.
That's incredibly frustrating, biologically speaking.
It happens in waves. Fever spike immune response, coat switch relapse. Fever spike immune response, coat switch. It's a relentless cat and mouse game that the patient eventually loses without treatment.
Deviously brilliant. Okay, let's leave the microscopic drop of water and zoom out. We're moving to the metazoa, the helminths, the worms.
The plate of spaghetti. Welcome to the world of multicellular parasites. These things have organs, they have nervous systems, they are animals.
And our taxonomy here is essentially dividing them by shape and cut.
Shape and cut is a great way to put it. We have the roundworms, the nematodes, and the flatworms, the platyhelminthes.
Let's start with Group A, the nematodes.
These are your classic earthworm-looking things, though they range from microscopic to macroscopic. They're cylindrical and tapered at both ends.
And their digestive system.
They have a complete digestive tract, mouth at one end, anus at the other. It's a tube within a tube design. This makes them fairly advanced compared to the flatworms.
And sexes. Do they have males and females?
Separate sexes. You have distinct male worms and female worms. The males are usually smaller and have a curved tail for holding onto the female.
Let's hit some clinical examples from the Jowett supplement.
Enterobius vermicularis, the pinworm. This is the most common worm infection in the U.S. and Europe. Mostly affects kids.
And the diagnosis is the famous Scotch tape test, right?
Yes, the Scotch tape test. The female worm lives in the colon, but at night, she migrates out of the anus to lay eggs on the perianal skin. The glue she uses causes intense itching.
So you literally just use tape.
You take a piece of clear tape, press it to the skin in the morning, and look at it under a microscope. You'll see the eggs plain as day.
Simple, effective, and gross. What about Ascaris?
Ascaris lumbricoides, the giant roundworm. These look like thick earthworms, maybe 20 to 30 centimeters long. You get them by eating eggs from contaminated soil, but their journey is wild. They hatch in the gut, but they don't stay there.
Where do they go?
The larvae burrow out of the intestine, get into the bloodstream, go to the lungs, crawl up your windpipe, and then you cough them up and swallow them back down.
Will they do a lap through the lungs?
Yes, a lung migration. This is biologically essential for their maturation. But clinically, it can cause a weird transient pneumonia called Loeffler's syndrome. Your eosinophils go crazy in the lungs.
And then they settle back in the gut and grow into giant worms.
Correct.
And hookworms, ancylostoma and neckhater.
These are the vampires. They have actual teeth or cutting pleats in their mouths. They latch onto the intestinal wall and suck blood constantly.
So the main clinical issue here is anemia.
Yes. Severe iron deficiency anemia.
Yeah.
And they don't wait to be eaten. They penetrate bare skin. If you walk barefoot in contaminated soil, the larvae drill right into your feet.
Ground itch is the sign of entry, right?
Exactly. Ground itch at the site of penetration.
Okay, that's the roundworms. Now, Group B, the platyhelminthes or flatworms.
And we split these into two subgroups, the flukes, which are trematodes, and the tapeworms, which are cestodes.
Let's start with the flukes. Trematodes.
Think leaf. They are flat, fleshy, leaf-shaped worms.
Their digestion.
Incomplete. They have a mouth, but no anus. It's a blind sac. Food goes in, is digested, and waste comes right back out the same way.
Efficient, I suppose. And sexes.
Generally, they are hermaphroditic, both male and female sex organs in one worm. But, and this is a massive exception for exams,
There's always an exception. The schistosomes.
the blood flukes, schistosoma,
Mm.
have separate sexes. The male is this robust worm that has a deep groove running down his body, and the slender female lives permanently tucked inside that groove.
Like constantly.
Yes, it's called being in copula. They are a lifelong couple swimming against the blood current.
That's surprisingly intimate for a blood parasite. Now for flukes, there is one animal host you absolutely must remember.
The snail. Every single trematode has a snail as an intermediate host.
Yeah.
If the exam question mentions a snail, the answer is a trematode, period.
Snail equals fluke. Got it. Now the second subgroup of flatworms, the cestodes, the tapeworms.
These are the ribbon-like worms.
Yeah.
They're highly segmented. Each individual segment is called a proglottid.
And what about their digestion?
None. They have absolutely no digestive tract. No mouth, no gut, no anus.
So how on earth do they eat?
They live in the soup. They hang out in your small intestine, surrounded by your pre-digested food, and they just absorb nutrients directly through their skin. They're tegument. They are the ultimate freeloaders.
And how do they hold on so they don't get flushed out?
They have a head structure called a scolex. It's equipped with suckers and sometimes a crown of sharp hooks. They grapple into the intestinal wall and just let the rest of their body trail behind them, growing longer and longer by adding proglottids.
Jawetz mentions Taniacilium, the pork tapeworm. This one has a really scary twist.
It does.
Yeah.
Usually if you eat undercooked pork with a larval cyst in it, you get an adult tapeworm in your gut. It's gross, but usually not fatal. But if you ingest the egg of the tapeworm, say from fecal contamination, the parasite gets confused. It thinks you are the pig.
And what happens then?
The eggs hatch into larvae, burrow out of your gut, and migrate to your tissues to form cysts. Muscles, eyes, and the brain. This is cysticercosis.
Brain cysts.
Neurocysticercosis. It causes massive inflammation and seizures. It's actually a leading cause of acquired epilepsy in developing countries.
So eat the cyst, you get a worm. Eat the egg, you get brain cysts.
Exactly. Keep the life cycle stages straight.
Another tapeworm mentioned is Diphyllobothrium, the fish tapeworm.
This one is massive. It can grow meters and meters long. And clinically, it loves vitamin B12. It aggressively competes with you for B12 in the gut.
Leading to megaloblastic anemia.
Spot on.
Yeah, we've done Protozoa, we've done Helminths. Briefly, let's touch on section 4 in Murray, the Arthropods, the vectors and villains.
Right. Arthropods can be the disease itself or the carrier.
For the disease itself, we're talking about things like lice scabies.
Right. Scabies is a mite that physically burrows into the epidermis. Lice live on the hair. That's direct infestation.
But mostly in this context, we care about them as vectors.
The taxi service? We mentioned the insects with 6 legs. Mosquitoes for malaria and Aflaria flies for sleeping sickness bugs, for Chagas. But don't forget the arachnids with 8 legs. Ticks.
Ticks are famous for Lyme disease, which is bacterial, but do they carry parasites?
They definitely do. Babesia. It's a protozoan that infects red blood cells. Very similar to malaria, actually. But it's transmitted by the exodes tick heavily in the U.S. and Europe.
And one oddball group, the crustaceans.
Yes.
Yeah.
Tiny water fleas called copepods. You drink them in unfiltered water. They are the intermediate host for Dracunculus the guinea worm.
The one that you literally have to wind out of the skin on a small stick.
That's the one. Fortunately, it is almost eradicated globally, but biologically it's fascinating.
We are in the home stretch now. Section 5, high-yield physiology and transmission wrap-up. I want to synthesize some of this for the listener. Let's talk about reproduction strategies.
There is a fundamental difference between Protozoa and Helminths here that you have to know. Protozoa multiply in the host. You swallow 10 Giardia cyst, you could end up with millions of trophozoites inside you via binary fission.
But Helminths.
Generally speaking, worms do not multiply in the human host. 1 egg equals 1 worm.
So if a patient has 50 hookworms,
It's because they were infected 50 times, or 50 larvae penetrated their skin at once. They don't have babies inside you that grow into more adults inside you.
that is a huge distinction. That's why worm burdens usually build up slowly over time based on environmental exposure.
Exactly. The adult worms are essentially egg-laying machines, and a Scarus female can lay 200,000 eggs a day. But those eggs have to get out of the body into the soil to mature and infect someone else. They don't hatch inside your gut.
Are there any exceptions to this rule?
Always. Strongyloids is the big exception. It can perform what's called autoinfection, where eggs hatch in the gut and larvae immediately reinfect the same person. This allows the infection to last for decades and become completely overwhelming in immunosuppressed patients.
Another key physiological sign for Helminths is eosinophilia.
Yes, eosinophils are the specularized white blood cells that fight parasites, but specifically they fight invasive worms. You usually see high eosinophil counts with tissue-invading helminths, like Ascaris doing its lung-lap strongyloids or Schistosoma.
But you rarely see it with Protozoa, right?
Correct. Protozoa generally do not trigger massive eosinophilia. So if you see a high eosinophil count on a CDC, it's a red flag for a helminth.
Let's recap transmission quickly. The how.
Route 1 is ingestion, fecal-oral. This is your cysts, like Giardia and Entamoeba, and your eggs like Ascaris and the tapeworms, arthropod bite, the vectors, malaria, leishmania, sleeping sickness.
And Route 3 is direct penetration, the barefoot risk.
Right. Hookworm larvae, strongyloids, larvae, and schistosomes.
Wait, schistosomes penetrate skin?
Yes. The infectious form of the Cercaria literally swim in fresh water and burrow right through intact skin.
Note to self: wear shoes and don't swim in waters with snails. Okay, we have built the shelves, we have categorized the beasts. Now let's test if you, the listener, were paying attention. It is time for
Rapid recall.
I'm going to throw 5 questions out SBA style. Single best answer. I'll read the question, then our expert will pause so you can think and then give you the answer and the why. Ready?
Ready.
Question 1. A patient presents with frothy, greasy diarrhea. Stool microscopy reveals a unicellular organism with a suction disc on its ventral surface. To which taxonomic supergroup does this organism belong?
Take a second to picture that structure.
Okay, time's up.
The answer is excavata, specifically the clade Metamonada. The organism is Giardia. The suction disc is the dead giveaway.
Question 2. You are examining a helminth. It has a segmented ribbon-like body and a scolex with hooks. You cannot find a mouth or an anus. Which class does it belong to?
Think about the shape and the digestion.
What's the class?
Cestodes, the tapeworms. The key feature is the complete absence of an alimentary tract. They absorb nutrients right through their tegument.
Question 3. Regarding the apicomplexa clade like Plasmodium, what is the function of the distinct structure found at the apical tip of the cell?
The breaching charge.
And the function is?
Host cell entry. The apical complex acts like a drill to chemically and mechanically penetrate the host cell.
Question 4. Most intestinal protozoa are facultative anaerobes. Which metabolic form is responsible for survival in the external environment?
The bunker versus the soldier.
The answer is?
The cyst. Trophozoetes are the fragile feeding form. Cysts survive the environment.
Question 5. Which specific helminth group is unique in having separate sexes within the trematodes?
The big exception to the hermaphrodite rule.
And they are?
Schistosoma, the blood flukes. Other trematodes are hermaphroditic, but they live in copula.
How did you do? If you missed any, you need to check out the tables in Murray chapter 67.
Specifically, table 67.2 for classification and table 67.4 for transmission routes. They are your high-yield summaries for board prep.
We have covered a massive amount of ground today. We've built the taxonomic scaffolding.
We have. We separated the water drops, the protozoa, from the helminths. We looked at how they move, how they eat, and how they trick our immune system.
Next time, we will start filling these mental shells with specific diseases in much more detail. But before we go, I want to leave you with a thought.
A provocative one, I hope.
Always. These parasites, especially the helminths, have been with us for millions of years. They evolved to live inside us for decades, actively modulating our immune system to prevent being rejected.
Right. They dial down our inflammatory responses. They are old friends in an evolutionary sense.
So in a modern world of skyrocketing autoimmune diseases and severe allergies where our immune systems are often just overactive, could the sudden absence of these old friends be part of the problem?
The hygiene hypothesis. It's definitely something to think about.
Thanks for joining us on this deep dive. Keep reading alongside the text, and we'll see you next time.